Hi, everyone, and welcome to BioVCN's Python Lesson 9. Today, we are doing, for the first time, a lesson that parallels that in one of the other channels. So this is meant to complement the Amplicons lesson on Ordinations Part 1 that the folks in the R channel put out for Amplicons. And we're going to be going over today how to specifically do beta diversity analyses in Python using Python pandas, as well as the Python scikit bio libraries. And before we get started, I want to take a little bit of time to actually go over what alpha and beta diversity even are to begin with, what those metrics are, and how we go about actually understanding that information. So to do that, I'm going to pull up our README. This I put together based on my own experience. So when I started working with Amplicon data and I was using pipelines and looking at all of this data output, I was so overwhelmed by the sheer amount of information, and I didn't even really know what I was looking at. What does it mean? How do I use these things to even understand my data? What's appropriate for my research questions? So I want to share that with you. So the first thing I ask is, what even are alpha and beta diversity? So these are two broad metrics that are used in bioinformatics. Alpha diversity mostly addresses the questions, how species rich or species uniform is a sample, whereas beta diversity is more answering the question, how different in its composition is one environment or sample compared to the others in your survey. Within each of these broad topics, there are also still many metrics, and it can be very overwhelming or confusing. And today I'm going to be focused on beta diversity metrics. Some beta diversity metrics, as with the alpha diversity, require your phylogenetic tree. So I'm going to reserve those for when we actually tackle alpha diversity. So I'm going to focus my attention on the beta diversity metrics that don't use a phylogenetic tree for today. So these include the two most common are the Bray-Curtis dissimilarity and Jaccard distance. So a Bray-Curtis dissimilarity takes the relative abundances of your species into account. So, if you think of an OTU table and you have, varying OTU counts per sample, relative abundances vary. The actual measurement itself ranges from 0 to 1, with 0 meaning the relative species abundances in two samples are exactly the same, and 1 meaning they're totally different. And of course, most fall somewhere in the middle. The Jaccard is similar, but it doesn't take relative abundance information into account. And this means your OTU table is essentially treated in a binary fashion. Is something present or is something absent? It also ranges from 0 to 1, 0 meaning two samples have exactly the same species makeup, and 1 meaning that they have nothing in common. So typically, these measurements are used on all combinations of your samples from your survey in an OTU table, and the resulting 2D data set you get is called a matrix, so X, Y, and that's, the first output. So then you have this giant data table of numbers, and you think, okay, well, what do I even do with this? Once these distance matrices are created, it's very common to perform some sort of ordination analysis to understand and reduce that data. And there are many of these. I've listed some of them, but the one we're going to be focused on today is principal coordinate analysis, PCOA, also known as multidimensional scaling analysis. It's a little hard to wrap your head around this concept initially, but what we can think about is if we have that Jaccard or Bray-Curtis matrix, and it's this collection of all these data points. If you were to fit a line to that collection of points, and that most minimized the average square distance from any of the points to the line, that vector would be considered your first principal component, and that represents basically the greatest variance in your data, typically what's supposed to be the most significant. And you kind of keep doing that over and over orthogonally until you go through many principal components. Typically, people are only interested in the first 2 or 3 because that usually contains most of the data variance. But the idea is that your data set's complexity is greatly reduced, which really helps look for trends, correlations, and things like that. And usually once you produce that PCOA, that transformed data, you visualize it in some kind of principal component plotting space, usually with your metadata, so that you can kind of qualitatively understand if any of your metadata variables might be driving some of the clustering that you're seeing. I know that's a lot to take in if this is your first time, but hopefully it'll be more clear once we go through the tutorial. So in this lesson, what we're going to do is we're going to clean up our OTU and our categorical metatable as we've done routinely in the last lesson. We're going to create our distance matrices. I'm also going to then show you how to perform a Mantle test, which just shows you if 2 matrices are or are not significantly correlated with one another. We'll then perform our PCOA on both of our matrices, and then I'll show you how to visualize that and color code those plots with the metadata. So with that, I'm going to launch the binder. In the directory here, we see the Python notebook input files. So let's open up the notebook. Now, there's a lot of libraries to import, but they're mostly plotting related, Matplotlib, Seaborn, and some sort of package from scikit-bio as well as pandas. So I'm going to import that, and then I'm going to import my OTU table and my metadata table. Now note that the metadata table, because for me this was formatted for Chime, it's a TSV. So if you're going to use the Pandas CSV reader to open it, you have to specify that the delimiter is a tab, which I've done. Otherwise, it'll be all sorts of messed up. Now I'm only going to briefly go through the cleanup stages because we did go through that in the last lesson. So our pretty standard cleanup, I'm going to quickly do this. So I'm showing where my chloroplasts are, getting rid of them, showing all entries where I have less than 10 reads of the OTU in all samples, getting rid of that. And then, of course, we are going to have to clean up the metadata table next. So this is, again, exactly like in the last lesson. I'm iterating through the samples the sample IDs that make those metadata indices. And I'm just saying, if it's not in my OTU table, if I don't need it, get rid of it. And this is actually really important for when you're doing the distance matrices and the plotting, because if something is missing somewhere, it will break. And then we see here blanks, not sequenced, great. We don't want our blanks to have anything. And then in my case, unfortunately, I had one sequence with an inhibition problem, but that's not for now. So we're gonna remove them. And then I am also showing here that you can see these all have non or not a number. So I'm gonna drop those. And again, I'm only rushing through this stage because it's entirely gone over in the last lesson. So please refer to that if there's confusion. Okay, so at this point, we're ready to start doing our analysis. For the scikit-bio matrices, we have to reformat our pandas data table into a numeric XY matrix or an array without any column headers or indices. So to keep track of what corresponds to each discrete sample here, I'm declaring sample IDs, which is equal to my column headers. Let's see what that looks like. So indeed, it's a list of all my sample names. Now, the next step is to transpose our data frame because we want our sample IDs to become our row indexer, and we want our OTU IDs to form the column headers. And this is because we need it to be in the same format as the metadata. So I'm going to do this using the pandas function transpose. And notice that I've done not just transpose, but I've said, okay, my data is my transpose OTU data dot values. And this makes it into just the numeric matrix. So it's going to be returning just the data values, not the column headers or the row indices. So now if we look at OTU data, we see that it's purely an XY collection of numeric data. Okay, great. Now our data is in the correct format and we have our sample IDs. So we can use the scikit-bio beta diversity function to create both types of distance matrices. There are actually many different kinds of analyses you can perform with the beta diversity function. So I've included a link to the scikit-bio documentation for the latest version of scikit-bio if anyone is interested in exploring other capabilities. So first, let's make our Jaccard distance matrix. So here, our setup is I'm using our beta diversity scikit-bio function, and I'm calling the method Jaccard in quotations. And the inputs are our OTU table and also the sample IDs themselves. Because otherwise, you know, without column headers, you can't keep track of what's what. Now, notice this warning. It's not a bad thing. It's warning you of what I mentioned in the markdown, which is that to do your Jaccard distance, it's converting your OTU table to a Boolean format. Is it present or is it not? And the nice thing is by building that in for you, you don't have to convert it into a Boolean matrix first yourself. but it's just making you aware. Now, conveniently, the Scikit Bio package, because it's outfitted with all kinds of seaborne stuff, if I were to just type in my distance matrix, it will just show up as a seaborne heat map. So let's check it out. So this is very busy because it's showing us literally every combination of samples against one another. So anyway, my point is you can actually look at the matrix qualitatively doing this. So now I'm going to do the same thing with the Bray-Curtis matrix. And you can see that the distribution looks very similar, but that there are differences, and that difference is the Bray-Curtis taking the relative abundances into account. So now I'm going to perform what's called a Mantle test. A Mantle test is common checkpoint analysis people use to compare distance matrices. Usually it's to make sure in instances before Scikit Bio that you haven't put a binary matrix into a Bray-Curtis and vice versa. So we do expect that these two matrices will have correlation with one another. That's expected, right? But it shouldn't be exactly the same. So the structure for that is it returns three things: the R, p-value, and n-value. So they're significantly correlated. The n value corresponds to our number of samples, and the R value tells us that while it is significantly correlated, they're not exactly the same. So we haven't screwed something up. Okay, so now let's recap. We've cleaned up our OTU data. We've cleaned up our metadata. We've created our distance matrices and sort of qualitatively looked at them in a heat map. We've performed a Mantle test. Now it's time to put our data through a PCOA. So here I'm going to use the Scikit Bio PCOA function, and it's really great. The only thing that you need to put in is the matrix itself. And then I'm setting a few other things. So this data frame proportions explained. You can access a really nice data table showing each principal component and the proportion of variance it actually explains by taking your results of the PCOA and doing dot proportions explained. So that's built into the Scikit Bio library. And then if you want the matrix itself, you can set that to the PCOA dot samples. So I'm going to run that. And again, it shows you these safety warnings. So for me, it says, warning, the result contains negative eigenvalues. Please compare their magnitude with some of the largest positive eigenvalues. For my case, I'm not terribly concerned because my negative value is really, really small and my biggest positive value is pretty big. But it's nice that Scikit Bio built in these checkpoints. And now let's actually take a look at what our distance matrix, our PCOA matrix actually looks like. So I'm gonna hit run. And this is what you get back. It's a data frame. So you have your discrete sample IDs that make up your indexes, and the principal components that make up your column headers. And I also want to show you proportions explained because this is really convenient. So I'm going to actually put it in here. And you get this really great table. So just looking at this, I go, okay, my first principal component is about 30% of my variance, 10% for the second, and so on. So it gives you a good idea of how many principal components are required from that table to fully explain your data. And now I'm going to repeat this for Jaccard. And let's take a look at that. So again, you get a similar data table. And let's look at the proportions explained. So you see it is slightly different. For example, so this actually reflects that Mantle test, that they're highly correlated, but they're different. Here we see in the Jaccard, our first principal component, 36% of the variance, right? Bray-Curtis, slightly different, 32. And another way to qualitatively check this is with the seaborne heat map. So what I'm going to show is a heat map of each one individually, and then I'm going to show a heat map of their difference. So here is the PCOA results of my Bray-Curtis. And also, again, you can see that pretty much once you get past the first 7 principal components, you can, I think, pretty safely ignore everything else just looking at the heat map. Now let's look at the Jaccard. So it looks pretty similar, but as we know, it's not exactly the same. And you can actually see here and here there are some differences. So now to qualitatively look at differences, let's look at a heat map of their difference. So again, this is just a qualitative check. Are they different? Have we done everything correctly? The seaborne heat map is super, super convenient for this. Let's see, we have a question from Haley. Does a negative value, oh, okay, Ben's already answered. Thank you, Ben. Sorry, I was busy plotting. Okay, so now here comes the kind of critical part. We've done all this work. Now we want to take our PCOA results and our metadata and use them together to analyze our data. Now, what I've done here is I've also looked for any columns in my metadata that are non. And the reason you must do this is if you're trying to do this kind of plotting and you encounter a column with non, it will break. It will not work. So I'm using my pandas function .isNA.isAny to find all of my columns with any nons in them. And I'm defining no non columns as everywhere where that condition is false, meaning there are no nons there. And then I'm setting cleaned metadata no nons to cleaned metadata no non columns. Now our metadata is ready. I do want to point out, and this may have been apparent in the last lesson, but I strongly recommend for this analysis, binning any continuous metadata you have into categories. It could even be numbers 1, 2, 3, 0, 1. Because if you have something like a continuous variable like chemistry, metal concentrations, it's really hard to see a meaningful cluster pattern with continuous data. So what I've done and what I would recommend to everyone else is I've actually looked at all my continuous metadata first as histograms, and I've binned them into the discrete number of bins required to fully explain my data. Because it's much easier to look at clustering with 0, 1, and 2 as colors versus a continuous spectrum of manganese concentrations, for example. Okay, so let's first look at the Bray-Curtis PCOA. I'm looping through and plotting, so I'm saying for every column in my categorical metadata, if the column is not equal to description, because that's like a weird text note column I had, take the PCOA and plot it. And so I'm passing in the metadata. I'm defining column as col because I'm looping through every column. So we're going to be producing a plot for each metadata column. My axes I'm calling 0, 1, 2. Just arbitrarily, they're the first 3 principal components, PC 1, 2, and 3. And then I'm setting my title to samples colored by, and then the column name. So now comes the fun part, and we're going to see what we get. Haley, to answer your questions, I did initially examine all the different rules to determine bin number to try to automate it. But in my personal opinion, I got the best results being, it's just one of those things you have to do manually, just make a bunch of histograms, sit there and look at your data, and make upper and lower boundaries, and then you can parse your metadata file and output a categorical one, which is what I did. It's just one of those things that requires some human interaction. But you can automate the creation of the histograms and binning of the data after. I just strongly recommend look at them yourself. Okay, so now I'm gonna look through all of these. So depth, size fraction, because I had 2 different particle sizes, sulfide. So it's taking your PCOA results and plotting them in principal component space. And what you're trying to do is really see if any of these metadata colorings correspond to a clustering pattern. It's a little hard to do this in the notebook because when you do this yourself externally, you can actually rotate the 3D plot and you can much better visualize that separation and clustering by being able to rotate it. But what I want to point out after I run this for Jaccard is we're going to see how some of the metadata variables and the clustering looks very different between the 2. So this is very important for something like, for example, sulfide or oxygen, where you expect that some species will be totally absent or totally present regardless of relative abundance data, because sulfide is toxic to them or because they're anaerobic. So let's actually look at dissolved oxygen. So I'm going to pull that up in Bray-Curtis, and then I'm going to pull that up in Jaccard. Actually, sulfide might be better because sulfide is very toxic to a lot of things. So we can see that the clustering is different, right? The PCOA Jaccard sulfide plot is not the same as the Bray-Curtis one. The Bray-Curtis one is more going to show you if there's anything interesting going on with the relative abundances of taxa being affected by sulfide, whereas the Jaccard is more going to tell you are things present or absent based on sulfide, if that makes sense. Let's find another good example. So maybe methane would be a good example. Let's pull that up. Okay, so again, here we have methane, right? Both matrices. The patterns are somewhat similar, but they're still different. And again, this is telling you different information about methane potentially driving the presence or absence of species versus how methane concentration might be affecting relative abundances of species. And these visualizations are more meant as a guide for you to go back to the pertinent metadata and do more extensive tests. So this is really kind of a qualitative approach to pointing you in the right direction for other analyses, if that makes sense. Okay, so that's everything I wanted to go over in the notebook. Thank you everyone for joining me for this lesson. And I really, really hope that it made your first grapple with ordinations and PCOAs a lot less overwhelming than it was for me when I first tried to wrap my head around it. I really tried to, for the markdown and tutorial, put myself in the shoes of someone doing it for the first time. And I really hope that I've explained everything in a helpful way. And I'll see you all next time.